The mapping of the human genome, begun barely 15 years ago, was an epic venture of discovery that opened up a vast new terrain for genetic research. Scientists, in their quest to understand the workings of our human genome, continue to create and to devise experiments that will reveal its truths. As words like DNA and genome have become part of the public discourse, and new words like transgenic species, cloning, and proteomics continue to enter our vocabulary, curators, artists, writers, and others search for a visual language to interpret, understand, and use the knowledge that ultimately affects every one of us. The exhibition photOGENEsis is an exploration of the discoveries, issues, possibilities, and promises of the genomic age as they are understood and translated by contemporary artists. It poses questions rather than offering answers, and invites reflection and response to the profound issues we face in the age of genomics. The years 2002-2003 marked the 50th anniversary of the discovery of the double helix, which spawned the genetic revolution and ushered in a new era of biology. First made visible by Rosaline Franklin through X-ray crystallography in 1952, the double helical structure of DNA was described by James Watson and Francis Crick in the journal Nature in 1953. Franklin's photographs are among the most important scientific images ever produced. Like Franklin's DNA photographs, scientific documentation for biological research has for years taken on an artistic form. Laura Albin-Guillot, working in tandem with her husband, produced early photomicrographs of crystallizations, plant cells, and animal organisms, which she then printed on metallic paper. Roman Vishniac, who was noted for light microscopy imaging techniques, produced early images of DNA strands. These images, magnified 25,000 times, suggest the beauty, if not the architecture, of our genetic structure. Vishniac's early photographs of nematodes, an important species for genetic study, seem timeless. Two scientists were recently awarded the Nobel Prize for their research into how genes control the death of cells in the development of nematodes. Helen Chadwick's photographs of human embryos, displayed like delicate jewels, are suggestive of stem cell research, that is, cells that are undifferentiated. Research using these cells promises future breakthroughs in the treatment of such diseases as Parkinson's. Felice Frankel's delicate flower is in fact a photographic representation of yeast, a simple eukaryote, an organism with a nucleus within the cell. Yeast was the first genome sequenced, and the 6,000 genes in yeast are remarkably similar to many of our genes and those of other higher organisms. Ellen Sandor, Keith Miller, and Dr. Kathleen Helm-Bychowsky's pH scologram shows actual DNA fingerprints that background the vertical strips of the DNA database that code for a small enzyme found in our tears and nasal secretions. This small set of sequences is but a fragment of the complex web of sequences that comprises every individual's elegant genomic map. We are in the midst of a revolution. It is being waged in the realm of digital information and genetic research. As suggested by David Palmer's painting, Shift in Faith, everyone's life view, be they computer savvy or scientifically illiterate, will be impacted in the age of digital and genetic technology. In an age of increasing specialization and complexity, artists who are able to understand and to interpret scientific information offer uncommon insights. These artists are scientifically literate, and their work elucidates and explores genetic research. Helen Donis-Keller, a professor of biology and art, mixes the idea of genotype, the genetic signature particular to each one of us, with phenotype, the impact of socio-economic conditions and environmental factors, to create a new form of self-portrait. The forces that forge our identity cohabit in a complex relationship of nature and nurture, reminding us, in the words of the artist, that kinship and diversity are related terms. Catherine Wagner hints at the dance within the cell that alters in microseconds in response to thousands of different circumstances in this series of images of magnified cells and cell division. Joan Fontcuberta's hemograms provide a mysterious abstract view of blood as a life form. A single drop of blood contains thousands of cells that carry the knowledge of your genetic inheritance. By introducing the gene for the green fluorescent protein found in a species of jellyfish into a few nerve cells in a living zebrafish, Reihardt Koster's photographs literally record nerve cells communicating with other cells. This cellular communication makes possible the function of all nerve networks. Further explorations by Ruiz and Altaba provide a convergence of science and art in his investigations of embryonic landscapes. These pictured here are a lowly fruit fly and a frog embryo. Nancy Burson's telomeres are the end-of-file sequence of human chromosomes and nearly gene-free. Telomerase, an enzyme, may hold one of the keys to an increased human lifespan, the microcosm impacting the macrocosm in ways which one can only imagine. Questions about Social Security, Medicare, and care for the aged are but a few possible issues. Looking back to the past in his use of the daguerreotype, physician Mark Kessell explores future questions about our genetic heritage and the features that make us human. Science both cures and kills. Better medicines and treatments will be discovered, but deadly organisms that can kill whole populations also will be possible. Living longer is a certainty, but what will the quality of life be? Prevention will decrease mortality, but what will life be like if the population doubles in 40 years to 12 billion people? Genetic defects may be eliminated, but what of creating new species? Hans de Neusser explores the preservation of life in his photograph of a frozen embryo. Louise J. Brown was the first child conceived outside a mother's womb. Now, with cryopreservation of a fertilized egg, children can be born after both parents' deaths. Mike and Doug Starnes' larger-than-life moth hints at the complexity of genetic programming. Environmental influences, such as artificial lights, can render genetic patterns maladaptive and cause moths to rush to a certain death, like Icarus flying too close to the sun. Genetic engineering produced the subjects for Catherine Chalmers' photographs and inspired Brian Crockett to create sculptural mice, each owning one of the 7 deadly sins. Scientists, in an effort to uncover the foundations of human disease, are producing thousands of rodents, genetically engineered to develop maladies from obesity to Down syndrome. Kunie Sigura's photogram provides an insight into the behavior of frogs under certain controlled conditions. Artificial illumination with the yellow spectrum can alter genetically programmed functions. Frogs suspend normal feeding and reproductive behaviors and may sit transfixed long after the light source has been turned off. Susan Kaprov photographically engineers a new breed of corn, blue-green algae corn. Most foods we eat today are the result of careful breeding and cultivation. Modern corn, or maize, has been cultivated over thousands of years from its wild relative teosinte. The artist says, "The question I ask is how much transformation will take place before these simple plants, known and eaten for centuries, become vestiges of a former world. As each individual's genomic map becomes known, personalized medicine will be possible. The focus will shift from treatment to prevention, and invasive surgical techniques, such as those shown in Max Aguilera Hellweg's photographs, may become unnecessary. Victor de Palma's graphic portrayal of an electric chair reminds us of the ways in which evidentiary DNA is changing the legal and penal systems. Evidence obtained from crime scenes decades ago can now be examined in a new way. More than 100 people have been exonerated and released from prison as a result of DNA forensic analysis. DNA provided the sole means for identifying 9/11 victims, as Richard Press's photographs remind us, a task that would have been impossible a decade ago. By creating multiple identities for herself with topical changes in hair, makeup, and clothing, Tomoko Sawada mirrors the capacity of the new science to alter species through gene line therapy. The 21st century is the century of genetics, where engineers will work at the cellular level and the engineer may be engineered. Transforming one's physical self and identity may become as easy as changing clothes, as Yasumasa Morimura's self-portraits suggest. The question of aging and living longer is explored in the work of Miyako Ishiuchi in his photographs of the 97-year-old Buto dancer Kuzuo Ono, and in the work of Miwa Yanagi, who asked friends to imagine themselves as grandmothers. From 1900 to 2000, life expectancy in the U.S. increased 70%, and genetic advances promise longer lives. Combining photographs and illustrations to construct a universal creature, Eva Sutton's software allows you, the viewer and user, to accelerate evolution and cause rapid metamorphosis. And Ellen K. Levy's digital prints of genetic and medical patents bring to mind the question, who owns your genes? Finally, Carrie Mae Weems explores some of the profound issues and possibilities posed in the genetic information age. Provocative, disturbing, yet seductively engaging, Weems' images float in a sea of sound. The viewer becomes part of the piece in the same way that her own life is touched by this ever-expanding knowledge of our inner universe. Science and technology have done much to improve our lives, but at the same time, they have the capacity to perturb our present, rewrite the past, and change the future. I know the artist's role is to provoke, and I welcome a consciousness-raising art, but I wish more of this work would convey the mystery, the elegance, the beauty of what the genome is about. The way in which this instruction book is put together, the dance within the cell that alters in microseconds in response to thousands of different circumstances and makes the right decision to keep that cell healthy, it's stunning. Mukiko Koie confronts the viewer with a visual composite of an ever-expanding world population. With advances in technology and medicine, many the result of genetic research, it is possible that there will be 12 billion people on the planet by 2040. As we get and beget, food, housing, and other resources will be needed in vast quantities. The genetic age offers ever-expanding hope, while at the same time raising ever greater fears. The knowledge that made possible the eradication of smallpox could be used in the wrong hands to decimate whole populations. In the name of security, genetic information and other technologies are increasingly used to identify and track individuals. Surveillance is increasingly a way of life, as shown by Miyuki Ishikawa. Penelope Umbrico appropriates the language of science for photographic arrangements of objects that she posits as coded biological messages. Artist Susan Rankaitis and dancer John Pennington conceived this artwork as a collaborative piece in response to biologist Robert Sinsheimer's writings on the human genome. By layering the codes, symbols, and visual clues of DNA language into her constructed images, the work reflects the complexity and intricacy, as well as the beauty, of every human being's genome, providing an appropriate backdrop for John Pennington's choreography. Michael Floman's fireflies provide a visual metaphor for scientific investigation. As Floman captures the magic of the midnight dance of fireflies in his photographs, the scientist isolates in the laboratory the gene that produces luminescence. Vladimir Manuel combines images of a paramecium, the double helix, and an ancient Khmer site with temple and palace ruins. Manuel derives his inspiration from my own dual admiration of science and of what science supplants. More than ever, we have the ability to decide who we want to be. We have more possibilities, more choices, and more liberty. The 21st century will open frontiers into the digital and biological worlds, genetics, bioinformatics, and nanotechnology. Clifford Ross's work suggests the vast and mysterious unknown, still open for discovery. The sea is the womb of creation, from whence all life forms issued. There are species and organisms beyond present knowledge, some existing in lightless and scalding sulfuric waters. The 25,000 pinpoints of light that illuminate Christopher Bucklow's human forms in his piece entitled Wedded Guests suggests the trillions of cells in every human being, each of which carries a complete copy of that individual's DNA. Chinese artist Zhang Huan inscribes the names of generation after generation on his portrait. In obliterating his own identity, Huan suggests the ancient source of human ancestry common to all Homo sapiens. And finally, the question of our origin may be reflected in Justen Ladda's Mirror. In the evolutionary divergence of lines, one led to the great apes and the other to Homo sapiens. From 97 to 98.5% of human DNA is the same as that in various species of chimps and apes. Thus, it is a small percentage of genes that are responsible for the gifts of language and poetry, of mathematics and logic, of curiosity and wonder. Scientists, in their quest to understand the nature of things, continue to create and to devise experiments that will reveal truths about the workings of the universe on both a macrocosmic and microscopic scale. The interpretation, understanding, and use of the knowledge, however, are up to all of us. This exhibition is an attempt to present art that demystifies the science, while at the same time suggesting the vast unknown sea of knowledge about our genetic heritage yet to be uncovered.